Hello everyone, my name is Rohit Babar, my roll number is 8 and today I'm going to explain you the topic of pharmacodynamics. So let's just go through what I'm going to teach you today. So first off we have principle of drug action, mechanism of drug action in which we are going to study enzymes, ion channels, transporters, receptors and finish it off with receptor occupation theory. So before moving on to this subtopics, I would like to explain you what the term pharmacodynamics is. So we have pharmacodynamics, which means which loosely translates to what drug does to body. So in more professional terms, it is the effect of the drug on the body. So pharmacodynamics is a study of the drug effects. It starts with describing what the drugs do and goes on to explain how they do it. So mechanism, it contains all the mechanisms and the theories which go through the drug action and mechanisms. So let's just straight go into the principle of drug action. So principle of drug action. So first principle is stimulation, enhancement of the level of a specific biological activity, usually already going on physiological process. So this is called stimulation. So the best example we have is how the heart rate is stimulated by the use of adrenaline. And then we have depression. So diminution of the level of a specific biological activity, which is like completely opposite to stimulation we can see. So for example, we can see barbiturates, which depresses the CNS system. So we have like that, and then we have replacement. So what replacement actually means is like if there is a natural hormone or enzyme which is very deficient in our body, so we supplement it, we replace it with the drug we use. So that is a kind of action or a principle of action that drug undergoes. For example, insulin, that is treatment of diabetes patient who used to take insulin injections. So that's the classical example of that. So next we have cytotoxic action. So we have chemotherapy, which includes like microorganisms, invading microorganisms, and and to kill the cancer cells. So that's all the principles of drug action which go through. We just go through them. So next topic is mechanism of drug action. So before moving on to explaining all the four big ones, so I would like to just take a moment and explain only a handful of drugs which act by the virtue of their simple physical and chemical properties. So here we have antacids, which neutralizes the gastric HCl, chelatic agents, that is EDTA, chelate the heavier metals like lead poisoning and all. So these are like the few examples, and so we can see these doesn't go under a intense mechanism of sort. They just act on their physical or a chemical property they have as a substance. So that's how very few drugs act like that. So the majority of the drug produces effect by the interacting with a discrete target molecule, which usually is a protein. Such mechanisms confers selectivity. So this breeds like selectivity in the enzymes and all that things. So functional protein that are targets of the drug action can be grouped into four major categories, that is enzymes, ion channels, transporters and receptors. So first we have enzymes. Almost all the biological reactions are carried out under the catalytic influence of enzymes. Drug can be either increasing or decrease the rate of enzymatic mediated reactions. So drugs can be both like increase or decrease the substance. Like here we can see in the image, like substrate when it receives on the enzyme side, it produces a product. Or the inhibitor when it occupies the receptor side, there is no product or a non-functional product is even formed sometimes. So these are like different types, and enzyme inhibitors, these are like two types. It can be competitive or non-competitive. Competitive inhibitors occupy the receptor side, while non-competitive do not compete with the substrate to occupy the receptors. So these are like the two differences between them. So let's go on to next study, ion channels. So what exactly is ion channels? How the mechanism is there? Like modulator, we can see here in the image modulator, blocker, opener, and like there is like a completely open ion channel as well. So these are like ion channels which are like gated. These are called gated channels. So they are like different gated channels in our body, like G protein gated channels and ligand gated. For example, ligand gated channel we have nicotinic receptors, and G protein we have cardiac beta 1 adrenergic receptors, which activates CA plus channels. As you can see, drug can also act on the voltage operated or stretch sensitive channels by directly binding to the channel, affecting ion movements through. That is, for example, we can see in local anesthetic there is like obstruction of voltage sensitive NFS channels. So the local anesthesia is given like that, and that's how it's done. So here we have certain drug moderate opening and closing of channels, like these like L type of channels sensitive for CA2 plus channels, and T type of channels sensitive to thalamic neurons. So these are like two drugs for that. So let's move on. So next we have transporters. So transporters are like membrane bound structures present on this cell membrane, obviously. It can either facilitate diffusion in the direction of the concentration gradient, or it can be a pump, like which pumps the metabolite or ions against the concentration gradient using the metabolic energy, that is the ATP. So that's how it works, and you can see in this picture we have transporters which is situated in between the membrane, and in the one transporter, first image we have the substrate binding to transporter, and another which has it leaving. So other we have leaving, and one there is also an inhibitor. So that is that, and that's how transporter works. And transporters are very different from receptors. Let's see what receptors actually are. Receptors are like macromolecule or a binding site located on the surface or inside the cell effector cells that serves to recognize signal molecule or drug and initiate the response to it, but itself has no other function. So receptors are like a communicator of salt from the outside of the membrane to the inside, and it has its effect inside itself. So that's how it works. And here we have receptors. So let's for explaining the receptor drug interaction, we have few terminologies. So I'm going to explain it now. So first off, we have agonist. So what is agonist? An agent which activates a receptor to produce an effect similar to that of the physiological signal. So we have agonists for like various systems which works exactly as the previous molecule, the naturally present one. So these are called agonists. Inverse agonist, an agent which activates the receptor to produce an effect in the opposite direction to that of the agonist. Same as like agonist, but produces a complete different, like on an opposite direction it acts, and that's why it's called inverse agonist. So next we have antagonist, an agent which prevents the action of an agonist on the receptor or subsequent responses, but does not have any effect of its own. So agonists are like they bind to the receptor site but do not have the same effect or any effect for that matter as compared to the agonists. So next we have partial agonists. So this is very interesting. It binds to the same receptor site, but it has a very sub-maximal effect, but like that. So that's why it doesn't quite get the same effect done. So that's why it's called a partial agonist. So these are like completely like agonists, but do not produce a lot of effect, but it's like sub-maximal effect. So that's it. So let's see receptor occupation theory. After studying qualitative aspect of the drug action, Clark in 1937 now produced a theory of drug action based on the occupation of the receptors by a specific drug and the pace of the cellular function that can be altered by the interaction of these receptors with the drug, which in fact are small molecular ligands. So like how the interaction is happening, he gave in 1937 a theory on it which is called receptor occupation, which actually deals with how the on the receptor how drugs occupy a space and produce a response, whichever may be, like how the metabolically it affects the cell. That's the theory he proposed, and the whole mechanism is explained. So that's subsequently it was realized that occupation of the receptor essential is very essential, but not itself sufficient to elicit a response. The agonist must also be able to activate the receptor. So that's the thing we want to learn. So before that, to explain the theory further, I would like to explain two terminologies here. The first terminology is affinity. Affinity is of a drug or receptor determines its concentration around the receptor required to form a certain number of drug response associations, which is called as DRS, drug response associations. Higher affinity implies that the same number of DRS will be formed at a very low concentration for the drug. But like if the affinity is less, then we will require more DRS. So that's the relation we have, the affinity, DRS, and this. And the second terminology I would like to explain is intrinsic activity. Intrinsic activity of a drug is a measure of its ability to induce a functional change in the receptor, which could be from 0 to 1, from 0 being nil to 1 being the maximum, or like which is like minimum to produce any response for that matter. So with these two terminologies, we can actually discuss the four terms we actually four terms I explained to you in the beginning. So the four terms mean, first is agonist. So what agonist has? It has a AI of 1, that's intrinsic activity has completely, that is maximum, but and as well as it has affinity, which is like binding property, and like if like in the lowest concentration it will produce a response, it will produce enough number of DRS. So that's the thing. Then we have comparative antagonists, have affinity but do not produce any intrinsic activity, that is their AI is 0. There has no production of activity, but there is affinity, so it competes with the agonist for the receptor side. So that's the according to the theory it explains. Then the partial agonist. So here we have, it has affinity but its intrinsic activity is below 1, so it doesn't produce a proper response, but it does have affinity and it's closer to the agonist than far from the comparative antagonist. So we have here partial agonist. So then we have inverse agonist. So inverse agonist, what it is it has affinity but its AI, that is the intrinsic activity, is in negative from 0 to -1. So it can be anything between 0 to -1, because it produces a response in a completely opposite direction. So that's how the theory explains all the four terminologies and the interactions of the drug and receptors. So now we are in the MCQ section. So the first MCQ is, what is the primary mechanism of action of the beta blockers in the treatment of hypertension? So option A, blockage of calcium channels. Option B, inhibition of angiotensin converting enzyme. Option C, blockage of beta adrenergic receptors. Option D, stimulation of alpha adrenergic receptors. So this was the first. And second, how do NSAIDs primarily exert their analgesic and anti-inflammatory effects? So option A, inhibition of COX enzyme. Option B, activation of opiate receptors. Option C, enhancement of serotonin. Option D, blockage of estrogen receptors. There was a second. So third, which of the following best describes the mechanism of action of the statins in lowering cholesterol levels? First, option A, inhibition of cholesterol absorption in the intestine. Option B, activation of lipoprotein lipase. Option C, inhibition of HMG co-reductase. Option D, stimulation of bile and bile acid production. That was the third. So we have the fourth. What is the primary mechanism of action of benzodiazepine in central nervous system? Inhibition of serotonin reuptake, enhancement of GABA receptor activity, blockage of glutamate receptors, activation of dopamine receptors. So fifth and finally we have, how do proton pump inhibitors work to reduce gastric acid and secretion in the treatment of acid-related disorders? Option A, inhibition of histamine release. Option B, stimulation of pepsin production. Option C, blockage of gastrin receptors. Option D, inhibitor of proton. So here we have all the answers. So we'll go one by one. So first question was, what is the primary mechanism of action of beta blockers? So the answer is inhibition of the beta chain. Yes, option C. First question, option C. Second question, how do NSAIDs primarily exert their analgesic and anti-inflammatory action? So answer is option A, inhibition of COX enzymes. So next is third, which of the following best describes the mechanism of action of statins in lowering the cholesterol levels? So answer is inhibition of HMG co-reductase. So that's the third question. Option C. Fourth, what is the primary mechanism of action of benzodiazepine in central nervous system? So the answer is option B, enhancement of GABA receptor activities. And the fifth and final question, how do proton pump inhibitors work to reduce gastric acid secretion in the treatment of acid-related disorders? So the answer is option D, inhibition of proton pumps. Thank you so much for watching. I hope you got something out of it. Have a nice day.